Ciclopirox is the most effective topical drug for the treatment of mild to moderate onychomycosis. Onychomycosis is a fungal nail infection affecting 12 to 13% of the population. Topical nail lacquer therapy is recommended for the treatment of mild to moderate onychomycosis to minimize the drug interaction and adverse events associated with systemic therapy. Two topical products are commonly used in most of the cases are Amorolfine 5% and Ciclopirox 8%, which are applied in an insoluble vehicle. Water-soluble Ciclopirox 8% formulation in hydroxypropylchitosan, called P3051, is an innovative product that does not require nail filing and can be easily removed with water. To compare the safety and efficacy of Ciclopirox 8% formulation in hydroxypropylchitosan, P3051, with Amorolfine 5% in patients with mild to moderate toenail distal lateral subungual onychomycosis. The study model was a randomized, controlled, parallel group clinical trial with a blinded evaluator. The blinded evaluation methodology was used to avoid the potential bias of an open-label design. Statistical analysis was performed using SAS 9.2 software. The Z-test was used to assess the difference between the two treatments. A total of 120 patients were taken for the study and were randomized into two groups. 60 patients were treated with P3051 and 60 patients with Amorolfine 8%. Patients with infected target nail area greater than or equal to 25 and less than or equal to 75% with positive KOH and culture for fungal nail pathogens at screening were included in the study. Patients with concomitant severe plantar tinea pedis, other nail abnormalities like psoriasis or lichen planus, were excluded from the study. Patients who were using other systemic or topical treatment were also excluded from the study. After a run-in period, the patients were randomized at a 1:1 ratio to receive P3051 or Amorolfine 5% nail lacquer. The patients with P3051 were instructed to apply the nail lacquer once daily for a 48 weeks treatment course and twice a week for Amorolfine patients, according to the instructions given in the leaflet. The patients were evaluated at 4, 8, 12, 24, and 48 weeks. No patients in either group had serious adverse events of severe intensity, and treatment-related adverse events that led to discontinuation of the treatment. The computed data for numbers needed to treat showed that only 3 patients with P3051 are required to get average 1 cure, whereas 11 patients with Amorolfine 5% are required to obtain the same clinical results. The number of patients cured at week 48 was 21, that is 35%, in the P3051 group, and 7, that is 11.7%, in Amorolfine 5% group. The complete cure rate at week 24 was 15% and 10% in P3051 and in Amorolfine 5% group, respectively. The treatment success parameter at week 48 was 58% and 26.7% in P3051 and Amorolfine 5% group respectively, whereas in week 24, 24 patients in P3051 group and 16 patients in Amorolfine 5% group were treated successfully. The p-value was equal to 0.0121 at this time point. At week 48, mycological cure was observed in all the patients in P3015 group and 49 patients in amorolfine group. At week 24, the mycological cure was observed in 58 patients in P3051 group and 52 patients in amorolfine group. The p-value at this point was less than 0.05 in favor of P3051 at week 24. Based on the above results, it was proved that P3051 is the most effective topical drug for the treatment of mild to moderate onychomycosis. Its efficacy is maintained in the long term and does not induce fungal resistance. The comparison of the NNT analysis indicated a better clinical efficacy of P3051 versus amorolfine 5%, ranging from 4 to 6 times.